Hi, and welcome back to the EDGE tutorial series. This video details the assembly and annotation module in EDGE. After this tutorial, users will be able to choose the appropriate assembler for their data and determine the best annotation tool for the assembled genome. As a reminder, we are running two job submissions in parallel. The first uses E. coli FASTQ files found in the PublicData testData directory. The second uses data from the SRA run SRR 2164314. Assembly algorithms are applied to raw data in order to align and merge reads to reconstruct the original sequence. As a result, thousands to millions of input reads are assembled into one or more long sequences called contigs. The assembly module is turned on by default in EDGE. As you will see in the interface, EDGE provides three different de novo assembler options. Choosing an assembler depends largely on the sample and time available for the analysis. IDBA performs well and fast on isolates and metagenomic samples. It's not ideal for large genomes. SPADES performs well but is computationally intensive. It works well on isolates. It's not ideal for large genomes. MegaHit is a fast solution for large and complex metagenomic samples. I'll quickly mention here that SPADES assembly allows users to input both short and long read data. So this is the module to accept PacBio or nanopore data. The built-in parameters for each assembler perform well. If you're interested in changing these parameters, go to the third-party tools section in the Read the Docs where you can locate publication information for each of these peer-reviewed tools. To note, in order to annotate a genome or perform any downstream contig-based analyses, assembly must be complete. There is an option to bypass assembly and use pre-assembled contigs for users that possess a FASTA file with this information. After assembly is complete, annotation can be applied to add biological information to assembled sequences. EDGE has two annotation tools to choose from. PROCA is appropriate for most cases. It's a rapid annotation of prokaryotic genomes tool. RAST will allow you to transfer annotation from a reference to a new genome. Let's pop over to the live demo. Starting with the E. coli sample, let's scroll down to assembly. Click to see the drop-down options. Under assembly, let's select SPADES since this is an isolate. We will leave all the parameters set as default, including the validation aligner. The validation aligner here is bowtie 2. It can either be set to bowtie 2 or BWA. They perform equally and are available if users have a preference. Bowtie 2 is selected by default. Click over to annotation. All of these settings are correct. Yes, we want annotation. We will leave a minimum contig length of 700. PROCA will provide annotation information. We want this done for bacteria. Let's update this module for the SRA data. Scroll down to assembly and annotation. For this sample, IDBA and MegaHit are both appropriate options. We will go ahead and keep IDBA selected as default and leave all of the parameters at their present values. Likewise, we can click on annotation. Take a look. We're going to keep everything here as default too. Okay, assembly and annotation are ready to go. In the next video, we will look into reference-based analysis.